I just want to thank everyone for joining this webinar today on the topic of using exhale breath to investigate the gut microbiome. My name is Hugh Davis. I'm heading up the commercial team here at Alstone Medical for our breath biopsy research products and services business. I'm going to be introducing the company as well as the breath biopsy technology before passing on to my colleague, Dr. Nabitha Nagalingam, who's our principal translational scientist and subject matter expert on the microbiome, and she'll be presenting some relevant case studies. I'll then pass on to Dr. Rob Money towards the end, who's our head of clinical biomarker services, and Rob's going to chair and host the Q&A session. Okay, so before we get started, just some housekeeping items. If you'd like to ask any questions during the presentation, please use the Q&A button at the bottom of the screen. We will endeavour to answer them at the end of the presentation. If you have any technical issues, then please contact the Alstone team via the chat button, also at the bottom of the screen. And just to mention, there are different views available in Zoom that you may be aware of. There's an icon in the top right-hand corner of the Zoom screen where you can select gallery view. That will allow you to see both the slides and the speakers. So I think we also have a short poll that we'd like to pull up just before we get into the main body of the presentation. Yeah, so this is just where you can indicate what area of research you're working in. If you'd like to do that, it's a multiple choice, so just tick the relevant box. Okay. Yeah. So Alstone Medical, we're based in Cambridge in the UK. We have just under 200 employees currently. And for over the last 7 years or so, we've really been pioneering and developing novel products and workflows for the reliable collection, analysis, and testing of breath, really with the end goal to have non-invasive breath testing in every home. So we've got sort of three major units across the business. We invest internally in biomarker discovery and validation, using exhaled breath to develop tests for early detection of lung cancer and also precision medicine in digestive disease. Pharma, consumer health and academic partners can also access our technology through commercially available breath biopsy research products and services across a wide range of different application and disease areas. And then finally, we're also developing at-home tests, point-of-care devices with expanding functionality that we're releasing under our OMED Health brand, where we'd look to launch those tests ourselves or more typically out-license to pharma or diagnostic partners to take to market. So we're focused very much on the analysis of so-called exhaled VOCs or volatile organic compounds. These are low molecular weight compounds that can originate from both endogenous biological sources as well as exogenous sources. Many of these are volatile metabolites that are products of human or microbiome metabolism that can reflect patient phenotypes. They can originate from the lungs and the airways themselves, of course, relevant for respiratory diseases, but also can originate from other organs and tissues in the body, can be carried via the bloodstream through the alveolar and onto the breath. So really, it's a full body phenotyping technology, if you like. And below there, you can see examples of the types of molecules that we can measure with our platform. Molecules in classes such as aldehydes, ketones, alkanes, alcohols, which can be reflective of a range of different metabolic processes, oxidative stress, lipid peroxidation, inflammation, as well described in the literature. And then also molecules coming from the microbiome itself, which is the subject matter of today's talk. So things like short-chain fatty acids, but also a range of other molecules as well that we'll talk about. So at Elstone, we've developed technology and products for both breath biomarker discovery on the front end through our Omni workflow, as well as developing tests for clinical or even home-based deployment on the back end. In terms of our discovery workflows, we have untargeted approaches. Again, that's our Omni workflow, where we can profile a very large and diverse range of VOCs on breath to identify novel biomarkers. In addition to that, we also take hypothesis-driven approaches, including the deployment of so-called exogenous VOC probes or evoke probes that can be administered through different routes, either orally or via inhalation, before being metabolized by target enzymes, which release volatile reporters back on the breath to boost the sensitivity and specificity of breath analysis, for example, to target disease-specific enzymes. So that's the approach we're taking in-house for our lung cancer and liver test programs, but we can also make this technology available to commercial partners in other areas. In terms of test deployment, we're developing approaches for both in-clinic and at-home collection, and then analysis offline, for example, through a central lab, as well as point-of-care home-based solutions where handheld devices with integrated sensors linked to a mobile phone app can provide more of a real-time readout of VOCs for longitudinal monitoring and consumer-based applications as well. So why would we want to analyze the microbiome using breath? Well, first of all, the microbiome is known to produce a wide range of different volatile metabolites. These include things like short-chain fatty acids, which are bioactive and have a range of health and disease-related effects, products of aromatic amino acid metabolism, including indoles, phenols, creosoles, alcohols, and others. So it's well suited to measure these types of volatile molecules. The other thing I would say is that breath testing reports metabolic changes more in real time, as opposed to stool analysis, which really reflects what's happening in the gut maybe 48 to 72 hours ago, and also therefore allows us to avoid a lot of VOC evaporation. Breath VOCs can be concentrated from large volumes of breath. Breath is rather an unlimited sampling matrix, so that allows us to pre-concentrate those VOCs, providing the right level of sensitivity, really down to parts per billion or even subparts per billion in some cases. Probably most obviously, breath testing is fully non-invasive. It's a convenient sample for patients to provide, which means that we can do sampling pretty much anytime we want. And it also allows us to perform high frequency serial sampling, for example, in response to a probiotic, a nutraceutical, or a pharmaceutical, and see how those molecules are being produced over a time course. And that can also allow us to determine approximate gut location of the microbes producing the volatiles. And then finally, as I mentioned before, there's a potential to launch home-based sample collection and analysis methods through handheld devices and the like. But breath is not without its challenges. Everyone breathes differently. VOCs are also present in the environmental air that we inhale, and there's a vast range of VOC concentrations and chemistries that we have to account for. So Elstone Medical has developed tools and methods to really reliably collect and analyze breath that minimize technical variability, minimize the noise and background that can come from the collection equipment itself, but also the air in which the sample's taken, and then reliably analyze a very broad range of VOCs, and importantly, accurately chemically identify those VOCs. So we have an unambiguous identification. We know what they are, and we can then pursue them in follow-up validation and analysis studies. So this is our breath biopsy collection station. This is our core technology for collecting breath samples, mainly in a research setting for biomarker identification and validation purposes. It includes the receiver breath sampler that allows for a reliable collection of breath onto sorbent tubes that are contained within the device. There's also our Casper portable air supply that will filter background VOCs from the air and provide that in a line to the receiver that the subject will actually breathe. Obviously, if you don't remove that, you're going to get variable levels of background contamination, false positives and false negatives within the data. And then finally, the software that drives the whole thing in terms of allowing for breath monitoring and collection and patient data entry for subsequent analysis. So subjects will breathe into the mouthpiece onto the receiver that you can see there. It's a disposable item that we provide for each breath sample to avoid cross-contamination. It's normal tidal breathing. The software will learn the subject's breathing profile through pressure and CO2 sensors that are integrated into the device before activating pumps to collect the correct fraction of breath. That's why we call it breath biopsy, really drawing it down onto the sorbent tubes. We tend to work with the end tidal fraction of breath, which helps us eliminate the dead space from the breath, which can be more variable and less concentrated in VOCs. And then once a fixed volume of breath has been collected, which is typically 5 liters of breath across the 4 sorbent tubes within the device, that takes about 10 to 15 minutes. The sampling will then stop. And as I mentioned before, this allows us to capture a very large volume of breath, pre-concentrating those VOCs onto the sorbent tube so that we can detect those more biologically relevant lower abundance volatiles that may be present. And then once the samples have been collected onto the sorbent tubes, they're uncoupled from the device, end-capped, and shipped back to our breath biopsy lab in Cambridge for analysis. And that's described in more detail on this slide. This is our Omni workflow. Once the samples are received by our lab, we will log them, dry purge to remove the moisture, end cap them with brass caps, and then they are stable for longer periods of time until we need to analyze the samples. We analyze using the gold standard thermal desorption GCMS analysis. We use high resolution, accurate mass, Q-exactive Orbitrap instruments from Thermo Scientific. These are exquisitely sensitive. They have high dynamic range and resolution. So it allows us to profile a very large number of VOCs from a given sample. Once the data has been collected, we can then deconvolute, extract the features, normalize the data, and then provide that processed data to our biostats team, who can perform various statistical analyses, univariate, multivariate. Importantly, we also match the data against our high-resolution VOC library so that we can have a high-confidence VOC identification assignment, which then, of course, allows us to understand more about the biology, but also then track those VOCs in follow-up studies, as well as match to our breath VOC atlas, which we've been building over the last couple of years. We're very excited about this approach. It's basically a searchable database of chemically identified VOCs. We've got a growing list of over 200 entries that we've shown that they exist and proven to be on breath, differentiated from background contamination, also with quantitative data from previous studies, including cohorts of healthy and disease subjects. So this has a number of benefits. Obviously, it allows us to understand biological pathways. It allows us to do in silico experiments, cross-validating results across different studies to accelerate biomarker development. There's also links to peer-reviewed literature on individual VOCs to learn more about them. And because we also capture the methodology in here, we can also learn more about what's the best method for faster optimization of VOC analysis platforms and assays in the future. In addition to the breath work, we also perform in vitro headspace sampling. We also offer this to customers that are interested in this type of analysis more in the preclinical phase. So here we can analyze cell culture, media. We can look at stool samples and various clinical biofluids that can be collected at the customer site, shipped to us, where we can then perform headspace analysis. We use so-called high sorb probes, which can absorb the volatiles from the sample, and we can process that on a liquid handling robot. And then the workflows on the back end are very similar to what I described previously using thermal desorption GCMS. Molecules that we find here, we can also look back in our atlas to see whether they are truly on-breath compounds and molecules that we'd like to pursue in follow-up clinical studies. And very briefly, we've also been working with the pharmaceutical company Boehringer Ingelheim on the development of a breath sample collection method that we can also make available. It's based on a modified flex event system, which allows for the collection of breath from intubated mice, a very clean breath sample from the lung environment. And again, captured onto sorbent tubes, which allows the same sort of workflow that I've described previously for human breath analysis. So just so you can understand a little bit more about how we work, the collection station itself with the receiver breath sampler will be based at the research site or clinical site. We can provide multiple stations if there are multiple sites involved in a clinical trial. We then provide a regular supply of kits, which include our breath biopsy cartridge, that's the sorbent tubes that I referred to previously, and the mouthpiece for each sample that's being collected. We train at the site, they collect the samples, they provide the sorbent tube containing the VOCs back to us. It's just shipped at normal ambient temperature, no need for dry ice or anything like that. And when they arrive at our lab, we then put it through our workflow, TDGCMS, stats and analysis, providing a report back at the end. Just to mention again that, we do have capabilities within the business. We have an engineering team, sensor development teams that can develop breath tests for more routine clinical use from biomarkers that we may discover through our workflows. Obviously, the appropriate test design will depend on a number of factors, including the concentration and chemistry of the VOCs being targeted, what are the user requirements, commercial requirements, etc. But we do have experience in this area. We've launched a CE-marked hydrogen methane digestive disease breath test that we offer up to the National Health Service and private practice here in the UK. That kit is sent to the patient's home for home-based breath collection. This obviously reduces hospital visits and supports diagnosis and treatment with expert interpretation of the results through our clinical partner, the Functional Gut Clinic in London. So those kits are sent back to us for mass spec analysis, and then we provide the results to the FGC, who then interface with the patient. So yeah, lots of different options here in terms of how we would go about developing a test. It could be a single-use home-based breath sampler with samples coming back to us for mass spec analysis, or all the way through to a handheld sensor for at-home collection and analysis of VOCs. And on that topic, we're about to launch our first version of a handheld point-of-care sensor, again for measurement of hydrogen and methane gases on the breath. This is a device that's connected to a mobile phone app, which allows us to review, or the subject to review, immediate results, as well as record food, symptoms, and other data within the mobile phone app that could be tracked against the levels of the gases on the breath. The data is sent to web portal for clinician review and a personalized treatment plan based on that data that's collected longitudinally. We're looking to launch that device towards the end of this year, early next year, and we've already launched a pre-sales campaign where people can sign up and express their interest. What's nice about this, of course, is that as we expand our capability and new VOC biomarkers are discovered, we can develop similar types of tests in the future based on this format or other formats. And with that, I'm going to hand over to Nabitha, who now walk through some specific case studies in the microbiome area.
Thank you, and thanks to everyone joining us today. We really appreciate your time and interest. In this next part of the presentation, I'll describe 3 approaches followed by case studies that illustrate how VOC profiling can be applied to microbiome research and biomarker discovery. But before this, I'll cover a little background on the microbiome for those who may not already be familiar with this area, although I suspect that many of you already are. The human microbiome harbors diverse microbes that play a fundamental role in our well-being. Beyond breaking down food to extract nutrients, our microbiota also plays important roles in educating our immune system, providing colonization resistance against pathogens, and in modulating key activities such as those related to the nervous system, to name just a few. Microbes exert this influence through the production of small molecules or metabolites. These metabolites can be products of our diet, our host cell, or microbial metabolism. They usually function in maintaining metabolic homeostasis, so disturbances to this balance can lead to disease. Some of these metabolites are volatile organic compounds that readily diffuse into the bloodstream. Here, they are transported to the lungs to be eventually released in exhaled breath. And these are the metabolites that we'll be covering. Some of these VOCs have been linked to health and disease. Here is a summary of some of such compounds with their microbial associates and where they function. You can see that these can be related to conditions that are remote from the gut. And these are only a subset of the conditions linked to the gut microbiome. Other key extraintestinal diseases that are also influenced by gut dysbiosis also include asthma and allergies. The links between the intestinal dysbiosis and disease continue to grow, and we as a scientific community are only just beginning to elucidate the complex role that the microbiome plays. Microbially derived VOCs can provide another dimension to investigate and gain insight into this relationship. Hugh has already outlined some ways that Outstone can support microbiome research. I'll delve into these areas further to describe how VOC profiling can be used, and these can be outlined in 3 approaches. First, in measuring the VOCs on breath, that can be interpreted either independently or integrated into a multi-omic approach. 2, using it as a tool to find novel associations, of which I'll also show an example. And 3, measuring VOCs from stool samples, which of course is a biosample that is routinely used in microbiome research. For the first approach, we can measure VOCs on breath using our Omni platform. We have amassed a library of reliably detectable VOCs, some of which are microbiome associated, as shown here. We can also see that these microbiome relevant molecules, which are short-chain fatty acids and products of aromatic amino acid metabolism, are measured on breath at levels significantly above background. Some other VOCs present in our Outstone VOC atlas are listed here. The asterisks indicate those compounds whose structures are depicted. Many of these compounds are derived directly from microbial metabolism, and some of them are indirectly associated with the microbiome. A case in point is benzene. It is not known to be directly produced by microbes, but linked to microbial metabolism. Studies show that Pseudomonas putida has metabolic pathways dedicated to compounds like benzene, toluene, and xylene. Thus, these can be considered microbiome-associated VOCs. This is a continuously growing list, as we are adding new compounds to the atlas and mining and curating the associated information, including the microbiome links. And speaking of microbiome links, we move to the second application. We can use associations to infer novel relationships between VOCs and the microbiome. In this example, we analyzed breath samples from healthy subjects. Here we used Outstone's internal library to investigate relationships among VOCs detected. After a clustering analysis, we can uncover some unexpected relationships. We postulated that 2 metabolites may be highly correlated if they are produced or metabolized by similar metabolic processes or perform similar biological functions. Here we see that several VOCs are clustered. We focused on acetic acid and propionic acid as known microbiome products, and believe that these associated compounds may also be microbiome related. In fact, some of these compounds will look familiar from the previous slide. For example, dimethyl sulfide in the propionic acid cluster. This is a known microbiome byproduct of the propionic acid metabolic pathway of some bacteria. Using such an untargeted approach can allow us to get a wider view of both mammalian and microbial metabolism and how these may be interconnected. Our third approach at Allstone is investigating VOCs from stool samples. The fecal microbiome is often used as a proxy for gut microbiome, as a non-invasive way to assess the composition and infer function of the gut microbial community. Allstone is developing capabilities for VOC profiling of fecal samples using headspace analysis. And this slide shows the output of a small feasibility study. To the right, we see a representative chromatogram from one of the fecal samples. We were able to identify several VOCs in a highly reproducible manner. A subset of these is listed to the left and contains many microbiome-associated compounds. Many of the VOCs we are observing in the fecal headspace dataset also overlap with VOCs that can be readily measured on breath, indicating that breath contains relevant microbiome-derived compounds. Moreover, because VOCs originating from the gut appear quickly on breath, an analysis of exhaled breath could provide a real-time snapshot of gut microbial activity and composition. While Allstone is well-positioned to provide analysis for breath VOCs, the inclusion of fecal VOC analysis in gut microbiome research offers another tool for biomarker discovery. Moreover, when combined with microbiome sequencing data, VOC profiling can offer insight beyond either dataset alone. After covering these methods, I hope you can see ways that Allstone can support some of your microbiome research. I'll now describe some case studies to show how we at Allstone have so far applied breath biomarkers to gain insight into microbiome studies. In the first section, I'll describe how the Omni platform was used to investigate type 2 diabetes and exercise in separate studies. Then I'll describe a study involving changes in VOCs induced by a lactulose challenge and iron supplementation. And finally, I'll introduce some of the work we are doing in test development that could help stratify patients with liver disease. Type 2 diabetes is a complex disorder that causes blood sugar levels to become so high that patients often suffer from excessive tiredness and thirst. It is also linked with increased risk of eye, heart, and nerve problems. Insulin facilitates glucose uptake. So insulin deficiencies result in poor regulation of blood sugar levels. This poor glucose uptake can be caused either by cells in the muscles, fat, and liver becoming resistant to insulin, or, as depicted here, the pancreas not making enough insulin. This in turn impacts cell metabolism and homeostasis, drives oxidative stress, and results in dysbiosis. Research from Das and colleagues have showed a clear shift in microbiome composition in type 2 diabetes subjects compared to healthy. In our study, we were interested in assessing how well our Omni platform would perform to detect VOCs in type 2 diabetes according to published studies. Participants were grouped as healthy, pre-diabetic, or diabetic. We requested that they fasted more than 2 hours, and a subset of them fasted more than 8 hours. Today, I'll only show the data from 25 subjects who fasted more than 8 hours. We found 156 VOCs reached statistical significance. However, we approached this data cautiously, considering the small sample size we had, which may potentially result in false positives. Therefore, to identify significant VOCs that we believed are truly relevant to disease, we focused on the 96 VOCs that met 2 criteria. First, that the VOCs from the breath samples needed to be significantly higher than background. And second, that the VOCs had a consistent trend following disease severity. That is, increasing or decreasing from healthy pre-diabetic to type 2 diabetic states. I'll present some of the more interesting findings. Here we see an increase in microbiome-associated compounds that trend with disease progression: acetic acid, isovaleric acid, and 3-methylindole. Acetic acid is a known product of the gut microbiome, and this increase was corroborated by Fink and colleagues, who also saw an increase in type 2 diabetes subjects. Another short-chain fatty acid is isovaleric acid, also known as 3-methylbutanoic acid. It has a characteristic unpleasant foot odor, as it can be produced by skin bacteria metabolizing leucine. It is also present in foods such as cheese and soy. This compound was previously shown to be increased in subjects with gestational diabetes. And third, 3-methylindole, also called scatol, is also readily detected in stool samples and contributes to its odor. It is part of the tryptophan pathway that others have also linked to type 2 diabetes. Although 3-methylindole did not reach statistical significance here, we think this might change with a larger sample size. There are also some other VOCs that showed a similar increase with disease severity, such as methylpropanyl ketone. Again, this was observed by others, but the mechanism of action here is still unknown. Considering this increase in type 2 diabetes and reported increases in non-alcoholic fatty liver disease and ulcerative colitis, we can speculate that it may play a role in gut inflammation and metabolic disruption. Other compounds also showed this curious increase linked to disease progression, but roles are unknown. This again highlights the Omni method as a tool to uncover novel relationships. I'd like to note that one limitation of this study is that most type 2 diabetic individuals were already taking medication. Nevertheless, we were still able to distinguish the medicated treated type 2 diabetic cohort from the pre-diabetic and healthy control subjects using breath VOCs. Our second case study was in collaboration with Bruce Johnson at the Mayo Clinic. Here we investigated the VOC profiles after exhaustive exercise, typified by ultramarathons. The Ultra Trail du Mont Blanc ultramarathon represents an extreme test of endurance. It is a race around the Alps Mont Blanc in Western Europe, looping through France, Italy, and Switzerland. It covers 170 kilometers, that's 106 miles, and climbs 10,000 meters, that's over 30,000 feet elevation. We'd expect this extreme challenge to trigger unique physiological responses. We use this opportunity to identify and study markers of inflammation resulting from physical stress by comparing the VOC profiles before and after the race from 24 participants. Although blood biomarkers have started to be investigated, very few studies have explored the potential of breath VOCs as biomarkers to show metabolic changes induced by exhaustive exercise. And to date, no studies have explored breath VOC changes in the context of running an ultramarathon that can range from 31 to over 200 miles. Here, we found 63 VOCs reach statistical significance in the pre- and post-race comparisons and identified 16 of them using the breath biopsy internal library. Acetic acid was one of the compounds that increased significantly. We already knew that acetate is the most abundant short-chain fatty acid in the human body, and it has an important role in as a metabolic substrate. The diagram illustrates how excess acetate that cannot be fully absorbed by the intestine enters the portal vein and is taken up by the liver or released into circulation. In the liver, acetate can be converted to acetyl coenzyme A, where it can be used as a substrate for fatty acid synthesis or for use in the Krebs cycle. under extreme fasting conditions, the opposite process can take place and the liver can oxidize fatty acids to generate acetate. Several recent scientific publications have demonstrated that intestinally produced acetate is an important energy source during prolonged exercise. Upon further investigation, 6 other VOCs that significantly increased in post-running breath samples suggest other underlying physiological changes in exhaustive exercise. These VOCs suggest that involvement of fatty acid oxidation and inflammation, which aligns with studies using serum samples. These data also suggest possible gut dysbiosis as a response. Taken together, we show that exhaled breath can be used to identify volatile biomarkers associated with exhaustive exercise, and that these include VOCs that indicate microbiome changes. Before describing the next case study, I'll take a brief detour to describe one of our tests. This test was used in the upcoming case study. Hugh already mentioned the HMBT. It is widely used in clinical investigation to help inform on the cause of digestive problems. This service is offered in the UK by Ulstone, though a referral from a healthcare practitioner is required. It is a separate kit that is different from the breath biopsy omniassay. Here, the subject ingests a carbohydrate specific to the test and then provides a series of breath samples. This is done by blowing through a straw into a collection tube. Then the test is mailed back to Ulstone for analysis. The test is based on the observation that hydrogen and methane gases on exhaled breath are derived from intestinal microbes as products of fermentation of undigested substrates. And here we have lactulose, fructose, or lactose. The pattern of these exhaled compounds can indicate underlying causes of persistent digestive gut issues, such as small intestine bacterial overgrowth, intestinal methanogen overgrowth, or other forms of dysbiosis. This information, in turn, can help with managing the condition. In the next case study, we use this test in an unconventional way. This study was done in collaboration with the Functional Gut Clinic. We used the lactulose challenge employed in the HMBT kit, but we looked beyond hydrogen and methane here. The background of the study is that iron deficiency anemia affects over 1 billion people worldwide, and they can be treated with oral iron supplements or IV infusions. Unabsorbed iron can have unintended side effects like enriching intestinal bacteria that result in bloating, gas, and constipation. Hydrogen and methane are 2 gases that have been associated with IDA following consumption of lactulose. These gases can travel to the lungs and be exhaled and so detected on breath samples. Here, we aim to investigate if other exhaled compounds are associated with oral iron supplementation using a lactulose challenge. We investigated VOC changes caused by 28 days of iron supplementation in apparently healthy volunteers. Samples were collected at multiple time points before and after the lactulose challenge and pre and post iron supplementation. Using the SIFT-MS platform, we performed targeted and untargeted analyses. Here I'll show 4 compounds of interest that were detected in the targeted analysis: acetic, butyric, and propionic acid, and hydrogen sulfide. All compounds that have been linked to health and disease. One noteworthy point is that the collection time points are important as they approximate the location where the gases are being produced, thus location of microbial activity. 180 minutes, for instance, would indicate colonic activity. For the short-chain fatty acids, there is an increasing trend with time suggesting that the further down the intestine, the more microbial activity is occurring, as we'd expect. There is also a difference pre and post iron supplemented groups. that is between the gray and blue bars. This indicates that the iron induced a shift in microbial activity. Meanwhile, you'll notice that hydrogen sulfide is not behaving the same as the short-chain fatty acids. First, there is a significant difference seen at time 0 before the lactulose challenge, and this is likely due to the 28 days of iron supplementation. Second, we also see that at day 1, before iron supplementation, those are the gray bars, there is a decreasing trend after lactulose challenge. This is also observed by others and thought to be due to microbial competition for hydrogen produced by lactulose metabolism. Hydrogen and methane producing bacteria can outcompete sulfur reducing bacteria, as suggested by the inverse relationship among these products, with increase in hydrogen and methane production and a concomitant decrease in hydrogen sulfide production. This final case study was inspired by a recent publication where ethanol was measured in portal vein blood of subjects with NAFLD, non-alcoholic fatty liver disease, and NASH, non-alcoholic steatohepatitis. The latter was recently been renamed to MASH, metabolic dysfunction associated steatohepatitis. And I'll use MASH for the rest of the descriptions here. This paper not only demonstrates an increasing ethanol level with increasing disease severity towards MASH, but also observes that the lactobacilliaceae abundance followed this trend and correlates to ethanol levels. This is an interesting phenomenon, as high amounts of gut ethanol may reduce the beneficial effects of experimental drugs. As such, ethanol ingestion forms part of the diagnostic protocol for MASH and represents an exclusion criteria for clinical trials. However, microbially produced ethanol is not accounted for as a confounder. Others have shown that subjects with MASH have microbiome ethanol production up to 100 grams per day, independent of alcohol intake. Therefore, we believe that a diagnostic test for gut ethanol production would help to assess and control for this factor. It can potentially identify a subset of patients more likely to respond to therapy in clinical trials, increasing the chances to bring a new MASH drug to market. We started by exploring baseline levels of ethanol in healthy subjects. Because ethanol produced by our microbiome is usually low, we attempted to boost the signal by feeding the gut microbiome glucose. Glucose is metabolized to ethanol as depicted in the diagram. Subjects fasted prior to the study to reduce VOCs from diet being a confounding factor. 10 participants were given a glucose challenge, and breath samples were collected before and after the glucose ingestion. And these analyses were done using SIFT technology. Here we see the results from the 10 subjects. The diagram on the left shows that on average, ethanol and acetone can both be detected over ambient signals in this small study. This is the first step to selecting a potential breath biomarker. On the right, we see the individual traces of each subject over time. The peaks all appear relatively early, before 50 minutes, suggesting that the ethanol and acetone are produced in the small intestine. It also helps define the length of the proposed test and frequency of sampling. This is only the beginning of this exciting research, and I'd recommend attending the BBCon online conference to hear more from my colleague Giuseppe about this. Hugh will let you know more about how you can join at the end of the webinar. Since gut fermentation produces very low levels of ethanol, often less than 20 parts per billion, we cannot use the commercially available breathalyzers for this. They can only detect over 100 parts per billion. Allstone's breath sampling devices and analytical methods have the capability to detect in the parts per quadrillion range, so have the potential to develop a very sensitive test. Like many of our tests, the ultimate goal is to make them easily accessible and user-friendly, ideally allowing people to use in the comfort of their own homes. And we aim to do the same here. This slide summarizes the case studies just described. The first demonstrating the use of our Omni platform. The second showing how we've used our at-home test kit to ask broader questions beyond hydrogen and methane. And the final study introduces a burgeoning microbiome test for liver disease. And now I'll pass it back to you, Hugh.
Thank you, Nabitha. Yeah, so just a quick summary of capabilities that you've heard here today. So a robust platform for breath VOC collection and analysis. That's the core technology that we've described today. We can also conduct literature searches to propose putative biomarkers that we can pursue in more of a targeted approach to complement the untargeted broad-based analysis platform that we have. And that can be done both from human breath as well as in vitro, and more recently with animal models as well, particularly mice and rats. So we can perform semi-quantitation, but we can also conduct absolute quantitation with standard curves for robust measurement of these molecules on breath. And then there's a whole team that support the process, from study consultation, project management, through to training for collection of a high quality breath sample. And then on the back end, obviously the analysis, stats and interpretation of the data, and then the ability to cross-reference those findings against the VOC atlas for accelerated biomarker development and further contextual information. And then finally, on the back end, the development of point-of-care field-based solutions for decentralized clinical trials or screening applications out into the future, which is where we want to take this business. Next slide, please, Nabitha. And just, Nabitha already referenced this already, but every year we run our breath biopsy conference. This time it will be an online event taking place between the 31st of October and the 1st of November. This is not just Alstone presenting. We're inviting in subject matter experts and breath researchers from around the world to come in and present their research, and you can register for free at alstonemedical.com/bbcon. And we typically get somewhere between 600 and 800 people registering for that event. So you're very welcome to join if you'd like to learn more. And final slide, please, Nabitha. If you would like to apply this to your work, or you have additional questions that we can't answer today, you can reach me, Hugh Davis, on that email address there, hugh.davis@alstone.co.uk. I'm also covering the America region, and then Liz Crone, my colleague, is covering Europe and rest of world. So please feel free to get in touch. And with that, I'm going to hand over to Rob, who's going to host the Q&A session.
Thank you, Hugh. Thank you, Nabitha, for the wonderful presentation. We've got a number of really great questions. We've been trying to address them as we go along, but I think there's some that we'll have to address live. So I think for my first question, Hugh, I'm going to go to you. It was mentioned in your introduction that breath sampling could be performed at home. Can you elaborate further on what this might look like?
Yeah, I think for the core technology that we provide as a research service, that's the breath biopsy collection station. Right now, that is something that needs to be installed in a lab or a clinical site, because it's a supervised collect. But we are developing simpler ways of collecting breath, single shot methods, and point of care solutions that could be deployed more in the home without the need for a supervision or training. So that's what we're moving towards in the future. We've got prototypes that we've developed that we're starting to use in our own trials, but that's the direction of travel for us for the future.
Thank you, Hugh. This one's for you, Nabitha. What evidence is there that these kinds of compounds may be specific for specific bacterial species or even strains?
Yeah, that's a good question and challenging to answer because, as many of you know, there's a lot of functional redundancy in the microbiome, so that many strains and bacteria often share particular metabolic pathways to produce certain metabolites. So tracing back which strains are responsible for certain metabolites can be difficult. However, it's not impossible. We can employ the headspace analysis, for instance, with particular strains of bacteria, so we can identify which VOCs are related to certain strains, and with a multi-omic approach as well, kind of get a proximity of the strains that might be related to particular VOCs.
Thank you, Nabitha. Kind of related to this, this came up in the chat a little bit. We talked a little bit about human breath collection. Can breath biopsy analysis, is it limited to human breath collection, or can the same principles be applied to the use of in animal models? And maybe that's a question for Hugh.
Yes, absolutely. I did pull up one slide quickly on this. So we've developed a method for collecting breath from rodent models. It's using a modified flex event system that we developed together with the pharmaceutical company Boehringer. So that allows us to collect the breath from intubated animals and provides a very clean breath sample that can then collect the VOCs onto sorbent tubes and go into our standard workflow in terms of TD-GC-MS and analysis. So that is a method that we've developed. We're making that available now to third parties. As of yet, we haven't looked at ways of collecting samples from other species outside of rodents in a routine fashion anyway.
Thanks, Hugh. I'm going to stick with you on this next question. You touched upon this a little bit in your introduction, but can you talk a little bit more about the applications of this technology in the consumer health area?
Yeah, so I think our first example of that is with the hydrogen methane breath testing device. So right now we provide a kit to the patient's home. They take a carbohydrate substrate and then a series of breath collects to collect the levels of hydrogen methane on breath. Those samples come back to our lab in Cambridge. We perform the mass spec analysis and provide the data to our clinical partners, the Functional Gut Clinic in London, who then interface with the patient to make a diagnosis and treatment plan. Our future version of that will be, as I described, a handheld point-of-care sensor-based approach, which will be hooked up to a mobile phone so that subjects can log details such as diet and symptoms, breathe into that device that will collect then real-time the levels of the hydrogen and methane gases and allow for more of a longitudinal monitoring and correlating the levels of those gases, for example, with diet and symptoms over time, which will allow for a better treatment and outcome for patients in the future. So that's our first version of a consumer-based device, but still with a clinician oversight. The data will be loaded up into a portal that could be accessed by clinicians. But the same approach could be used for other VOCs in the future as well.
Thank you, Hugh. Maybe for Nabitha, we talked a little bit about VOCs and as disease markers. Can volatile compounds also indicate patient response to the microbiome and microbial metabolites, as we know?
Yes, absolutely. We think so. With microbiome interventions, whether it be probiotics, prebiotics, or fecal transplants that disrupt the microbiome or shift the microbiome, we expect to be able to monitor those shifts with volatile compounds. In fact, any kind of intervention, microbiome or not, drugs, for instance, we should be able to monitor those changes from the microbiome in our gut, as well as our host response.
Thanks, Nabitha. This next question is kind of, I'm going to make it more specific to short chain fatty acids here. We've had similar questions in the chat. How well do plasma levels of short-chain fatty acids correlate with breath short-chain fatty acids? And Hugh, I think I'm going to lean that towards you, given some of the discussions we've been having on the business development side there.
Yeah, there's there's not much in the literature around that today. Nabitha, please correct me if I'm wrong there, but we are planning a study and perhaps a number of studies where we'll have the chance to correlate the levels of short-chain fatty acids in blood and stool samples with breath. So those studies we are planning in the near future, but we don't have that data as of yet.
Great. And then I'll leave this to either you or Nabitha. Volatile sulfur-containing compounds have been reported as markers of a variety of diseases, like cancer and respiratory infections. Do you measure volatile sulfur-containing compounds in your assay?
We do. One example is dimethyl sulfide. I think Nabitha may have touched on that compound, but there's a range of others that we can measure as well. So I think the thing to do is, if you're interested in a particular compound or class of compounds, please let us know. We can first of all look into our VOC atlas to see whether it's something we've measured in breath in the past, but we can also give a good estimate of what's the likelihood that we'd be able to measure it in the future with a given method as well. So I think that's a conversation we'd need to have.
Okay, maybe one last question before we toss it over back to you, Hugh, for a close. I think this is a relatively quick question. How amenable is your breath collection method to high-frequency serial sampling in the same individual?
Yeah, I can happy to take that one. The short answer is it's very amenable. Obviously, breath is well tolerated by patients. It's much more difficult to ask somebody to give a repeated blood sample, and impossible, obviously, to do that with stool sampling as well. So with breath, obviously, you have got the ability to collect samples, I would say, at least every half an hour if you needed to, as part of any given study.
Great. Thank you, Hugh. I didn't know if you wanted to give a quick close on the webinar. I think we're almost out of time.
Yeah, happy to close here just to say thank you to everyone for joining and your interest in our technology. And again, if you do have any further questions or would like to discuss applications in more detail, please do get in touch with us.
Thank you.